Hello, I'm Adam Deardorff. Nice to see everyone. Special thanks to Dr. Novak, Marlon, everyone at Ohio Health, and ALS United for putting this together. I think it's been a really great event. It's a particular thrill for me to be here alongside of Dr. Neal and Dr. Kolb, both of whom have taught me and mentored me over the years, especially Dr. Kolb, who's helped mentor my scientific career as well. So a real thrill, personally, and also a real honor to be here with all of you, too. I think I speak for all of us when I say that we learn more from you living with ALS and your loved ones than I think you do from us. So it's a real thrill and privilege, so thank you very much. I thought for my talk, what I would basically do is kind of the how a bill becomes a law, but more like how an idea becomes a medicine. So we're just going to briefly go over what are the steps of developing a new medication. So drug development in the US is regulated by the FDA, and according to them, you can break down this process into 5 steps. The first step is the idea. So we discover new drugs in a few different ways. You can learn new insights about the way that a disease works, what goes wrong on a cellular level to cause a disease, and then you can target those cellular elements. You can repurpose existing therapies that might have effects that we didn't quite think of before. Or you can develop new technologies to target specific sites in the body or manipulate specific cellular processes. Gene therapy is a good example of that. And at this stage, this idea stage, there are thousands and thousands of compounds that might be candidates, but after extensive testing in the scientific laboratory, which is kind of around where myself and Dr. Kolb and Dr. Elbasiouny spend much of our time, we can narrow it down to just a few promising medicines that look like they could use some further study. Then we move on to what's called a preclinical study. So this is a study that we do to make sure that the medicine seems like it would be safe. So it's done not in people, but rather in model systems. We can do them in model systems that we call in vitro, which means in a test tube or cells that you've grown in a dish, or in vivo, which means in life, so living whole animal models we'll give the medicine to. Usually these preclinical studies are pretty small, but they give us detailed information on, is the drug really doing what we think it's going to do? Does it appear safe? Is it super toxic? And after all of these studies, we review the findings, and if we think this looks like a really promising medicine, we need to try it in people, we can file what's called an investigational new drug application to the FDA. And then we go on to clinical studies. So clinical studies, we can break down into 3 phases. And then after FDA approval, there is a 4th phase of clinical study that I'll mention too. The first phase of clinical research I like to think of as, is this medicine safe? So you take a small group of people who are either healthy or have the disease, you give them the medicine, you adjust dosing levels, you adjust the manner in which you're dosing, whether it's oral or an IV injection or something, and see, is this medicine causing unintended consequences that would make that medicine dangerous or just simply intolerable. Also in these, we're looking for how does it work in the body, and again, what's the best dose that we can give. Then we go on to phase 2 studies, and I like to think of this as, does it work? So you're looking up to several hundred people with the disease. You give it to them at the dose you determined was best in your phase 1 study. And we're always monitoring for safety, so see if there's any unintended consequences with a larger patient population. But also now you're looking at, is it efficacious or does it influence an outcome? So in ALS, you might think, does this influence respiratory function? Does it influence your ALS functional rating scale scores? Does it influence your ability to swallow, your ability to move? Because these are smaller studies, they're not really large enough to determine if the medicine is effective in changing outcomes, but if we see that there is some kind of efficacy, then we can move it on to what's called the phase 3 study, which is, does it change outcomes? So we're giving these to as many people as we can with the disease, just as we would in actual practice. And because these studies are larger and they're longer in duration, we can determine if the medicine is actually effective in changing outcomes for the disease. We can also, because it's a larger study, monitor for more rare side effects. Once we get through this study, this phase, we look at all the data. If we feel like things still look promising, you can submit it to the FDA for approval. The FDA has 6 to 10 months then to look at all of your data. They come visit your lab. They come look at everything that you've done. They review your notebooks. They say, did you do all of this correctly and with the most rigorous of research standards possible? And if you do, they can approve the drug. If the drug is approved, then it goes on to, that says phase 4, it should be phase 5, which is post-marketing review. And this is review of the medication after it hits the markets, where people are getting the medicine prescribed to them, their insurance companies are paying for it, they are taking it. It's the largest population of people that you could get studying or taking the medicine, and you're looking to see, are there any really, really rare side effects, right? Because even with the most rigorous phase 1, phase 2, phase 3 studies, you really don't get the true picture of a drug's safety until a lot of people are taking it, because everyone is different. So this is a long and expensive process, as you can imagine. But there's been some new developments in study protocols called platform trials. And platform trials allow us to test multiple investigational products in a lot of different participants at a lot of different study sites using what they call a common master protocol. So where you can see sort of in that top panel, sort of our traditional trial design is you've got one group of patients, you give them one drug, you follow them for a long time, and you see what happened. In a platform study, you've got one group of patients, and you give some of them drug A, some of them drug B, some of them drug C, some of them drug D. And you follow all of those out over time. If one of those drugs looks like it's not working, it's out of the study, and you put a new one in. If one of those drugs is looking like it's working, well, then you keep studying that drug. In ALS, the first and probably most famous and maybe only platform trial is the Healy platform trial. This study is being performed at over 70 sites across the country, so it's easier to recruit patients. Because you're doing the same study at a bunch of different sites, you can decrease the time it takes to study different drugs. We can share data across sites to help benefit the entire ALS community. By sharing infrastructure, having a master protocol, you reduce the cost of developing the drug. And, like I said, new investigational products can be added to the platform at any time as they're discovered. This is kind of an example of how the platform works. These are the first 4 regimens, which are A, B, C, and D. I think now we're up to regimen G in the Healy platform trial. But basically, you would, I'm pointing just to this screen, sorry for the people looking over there. You'd take your study population here, divide them up randomly into people who got regimen 1, or regimen A, regimen B, regimen C, regimen D. Then amongst each of those people, they were randomized on a 3-to-1 ratio of who would get the drug and who would get a placebo, which means not drug, but they think they're taking it. And you monitor them for the outcomes. And if any of them look promising, they could go into what's called an open label extension, where even after the study protocol ended, those who are getting the drug could continue taking the drug to follow it. And I think that's my last slide. Yes, and that's our Dayton ALS clinic. That's everyone in the clinic, including our MAs and our front desk staff and our office manager. And special thanks again to ALS United Ohio for making that dream a reality for us. So thank you very much, and I will turn it over now to Dr. Kolb, or Dr. Elbasiouny, excuse me.
That's the wrong one. Yeah, we just want to switch on the other one. There we go. That's good.
Up and running. Yeah, perfect. Well, I would like to start by thanking Ohio United for this meeting. you truly united Ohio today on ALS. So this has been really great. Today has been my first time to meet, with Dr. Neil and with Dr. Kolb like in person. So this has been really great. So I truly appreciate having all of us like in one place. I am Sherif Elbasiouny. I am a professor of neuroscience and biomedical engineering at Wright State University. My lab is an interesting lab because we have students from engineering, neuroscience, biology, different disciplines. So all disciplines are really represented in the lab. And this is because this diversity that we have, diverse student population that I have helped in investigating the problems or the projects we do like in a much greater level of detail. So before I talk about ALS, I wanted to give you some information about the lab. We are interested in movement and movement disorders. So one project that we have in the lab is on aging, why we become weak as we age. So this is something that we're studying. We study the changes that take place in the nervous system as we age with the goal of figuring out what we can do in terms of like at the cellular level to slow down this process. This is not going to prevent us from aging, but hopefully it will help us like age well. Another project that we have in the lab is on prosthetics. Where after people like lose a limb, they get fitted with a prosthesis. So what we work on is making this prosthesis feel and function like a normal limb. So basically, this prosthesis can be fitted with electrodes, can be with sensors, so that when an amputee make a touch on a project on an object, they can feel the object. And the same, when we want to move, when the amputee wants to move his or her missing limb, then the prosthesis would move naturally as if it's a natural limb. The largest project that we have in the lab is on neurodegeneration, primarily ALS, but we're also interested in studying frontotemporal dementia, which is also comorbid with ALS. Our approach, or one exciting feature of our approach is that we do human experiments, animal experiments, and actually we use the animal experiments to inform our human experiments. And that's very powerful because this saves us from doing like trial and error on human subjects. That's one. Number two, there are things that we can do in animals that we can do in humans. And I will show this in a minute. One of the primary challenges of ALS is that more than mutations in more than 40 independent genes have been shown to result in ALS. So there is a lot of heterogeneity in this disease. And with that, it becomes really very hard with a rare disease to find enough number of patients so that you have in a specific mutation that you can study and understand how disease pathogenesis or how the disease progression takes place. But that's the advantage of animal research, because with animals we can generate these mice with different mutations. We can study them in large numbers, enough numbers, so that we can understand what's happening in the disease. What's really very powerful as well is that we know from day one, when the animal is born, if the animal has the disease or not. And we know and we can measure from cells that are eventually going to degenerate before they start degenerating. With this, we have very powerful tool to be able to really see the changes before they start and track these changes during the disease process. I just put this slide like really last minute just to give you an idea about what the level of detail we can get into. So basically, we can remove the spinal cord, like the part of the nervous system, the spinal cord of the mouse, place it in a dish, and basically we can advance with electrodes and we can measure from these cells that are going to degenerate and measure their properties before they degenerate or during the degeneration process, which informs us a lot about the changes that they experience. This is another slide that shows the different approaches that we use in the lab. So we do something called electrophysiology, where we measure the electrical properties of the cells. We can also stain the tissue, look at the different level of the expression level of different proteins, and we can put all this information into computer models and run simulations and do machine learning. These are our advanced tools which allow us to look beyond the data. Because in a disease, when you measure from an animal, you see changes. But some changes are not important changes, and some changes are important changes. And if you were to develop an effective treatment, you want to target the effective changes, not the secondary ones. That's why many times you have a drug, and then it doesn't work. It's doing something, but it's not actually targeting the primary changes. That's why it's not effective. So with that, we try to minimize this trial and error by being smart and take the data before we go to humans and run and do like AI and machine learning to really understand what target we should be looking at. So 2 primary goals that I would like to share with you in the lab. One thing is drug discovery. So with all of the knowledge that we collect from animals, we kind of like, determine what basically drug targets that could be, we could use drugs to really target and see if they will slow down the disease. So we inject mice with these drugs, and then we look at the responses, and we see if the drug has a negative response or a positive response. If it's a positive response, obviously we do more investigation on the drug. If not, then if it's negative response, then we look at a different drug. So we do this like, I do not want to say like trial and error, because I just shared with you that we do it in a smart way, but we do it in mice, not necessarily in patients. And one of the challenges of ALS is that there is no biomarker for the disease. And it has been shown in research that if you start early in mice that have the disease, if you start giving them treatments early, the impact is much higher, and the likelihood of success is much higher in extending survival than waiting until symptoms emerge. Because by the time symptoms emerge, a large number of the cells have already degenerated. So one thing that we learned in the lab is that, with many of in the different animal models that we studied, is that there are changes that take place in the EMG signals as these cells degenerate. So we have information that we measured in animals, and now we're testing them to see if these biomarkers are also present in ALS patients. So with that, I would like to really share the news that we have a study that is basically looking into the development of a novel biomarker in ALS. And basically, this is like the setup. So this is not mice. This is in humans, in patients, where just simple leg contractions or arm contractions, and we're measuring EMG signals. These are surface EMGs, not needles EMGs. So there are no poking. There is no discomfort. Really, all like surface. And this is like the shape of the electrodes that we place. And this is the signals that we measure, surface EMG. And what is really cool is that through some advanced engineering approaches, we can reverse engineer from the EMG signal what is the firing of the cells in your spinal cord without really poking the spinal cord or measuring anything directly like in animals, because we can do this in humans. And this informs us of and this allows us to compare the data we get from humans relative to the data we collected from animals. So the study, we are actively looking for enrollment. It's open. The 2 visits that take place at our lab at Wright State University. We try to, we understand that some folks will be coming from Cincinnati or from Columbus, so we try to cover some, like gas and trip expenses. What's really interesting and exciting for me to announce is that with some collaboration now taking place with Ohio Health, Dr. Novak is our clinical consultant on the study, and Ohio United has kindly supported the study. So that's the QR code. So if you know patients that will be willing to enroll, please share that with them. And this is the clinical study staff. Bradley and Sharon both are in the audience. So if you have any questions, please ask them. And that's these are the nerds, like the larger lab. Well, Teresa Garrett is the lab manager. She's in the audience as well. and we are supported by NIH, Ohio Health, ALS United, as well as National Academy of Sciences. And thank you, and we'll be happy to answer any questions.
Can you go back to the QR code?
Oh, okay. It's no longer. Oh. Yep. Oh, thank you.
All right, I will have some QRS cards or whatever they're called also. Hi, everybody. I am happy to be here from the National Championship Ohio State Buckeyes, which I'm very, very happy about. I had absolutely nothing to do with it. Nevertheless, but my joke is always that if you really want to see strength at Ohio State, you should come to ALS Clinic. And so I'm very humbled to be here with you guys. Thank you to Marlon and Melissa for inviting me here, and for ALS United. They've always been such strong supporters for us. And this truly is a wonderful event for all of us as colleagues, and also just to make sure that the community here is all together. What I'd like to do today, and I'm sorry that I missed the morning sessions. We're actually hosting an ALS researcher from the University of Florida who's speaking today at Ohio State. And so I was there, so it's an ALS excuse, even when I was there. All right, if I can figure this out. So I always think about, the research for any, for our patients as part of this Buckeye tree. Where in the canopy of the tree is the clinic. That's where really the focus is. That's where the diagnosis is made. That's where the care is given. That's where the proactive, palliative mindset is there. But it is rooted in this basic science, and researchers like who we're hearing from today, who may not be directly seeing patients, and you may not even meet, but who are working their asses off trying to figure out what the hell is going on with ALS. The reason we don't have a treatment for ALS is because we don't know what the hell is going on with ALS. We don't know why. We don't know why it is that those neurons that are connected to your muscle start to die while all the other neurons in your body seem to be fine, and so are all the rest of your cells. That is the science nut to crack, and that is where I think that we're very passionate about, and that's why this sort of team approach is good. What connects the clinic to the scientists is this translational research. We call it translational because you starts in the clinic, goes around towards the laboratory, and then comes back up. And so I'm going to talk about what's going on at Ohio State. I think you'll all be real proud of what's happening right here in Ohio that's making a difference in what is happening not just nationally but internationally in the ALS community. I just want to give a quick shout out. Of course, our multidisciplinary team, just as wonderful as the one here at Ohio Health. We all work well together and sometimes see the same patients, but it's a privilege to work with this team of people. So we have maybe 11 providers in one room who are going in, and we try not to go over 2 hours. I remember that 2 hour thing, but sometimes we'll do that. Just a shout out to the whole team there. One of the things that I think Dr. Novak may have talked about is this notion of palliative care. One of the things that I'm happy to announce is that we have a new division of neuropalliative care at Ohio State, which is really one of the first of its kind. And Dr. Elise Everett has joined us from Washington University in St. Louis, another prominent research-based ALS clinic. And we are going to be, not only is she just transformative and trained in a different way than neurologists and the rest of us, but she is also going to be contributing towards our research mission as we move forward. So enough about that. Let's focus on the research. So here's my sort of attempt at going from what Dr. Deardorff was talking about here in the clinical trial side. That's the investigator-initiated clinical trial part. That's what goes back into the clinic. But also, how do we go from the clinic back into the labs? And that is what Dr. Elbasiouny was so wonderfully illustrating as far as how that goes. This is how it's all connected. It starts from the patients. From patients, we try to understand things that we can take to the lab and study. And as Dr. Elbasiouny told us, one of the things that is really transforming research in ALS is the genetics. It is not just a thing anymore that there's family history and no family history. It is a genetic disease most likely, and as our technologies to understand what's happening in the genome improve, we continue to uncover more and more genes that are associated with ALS. And I'm happy in the discussion we can get a little bit more granular about that. But on the left here are people with a family history, and you can see that there's about 75% of people who have a family history of ALS, we have identified genes for that. But there's still people who have family histories, they have ALS, and we don't know what the gene is. But here's the big deal, is that the 90% of people who do not have a family history, we increasingly find those genes that are mutated in those people as well. And that is why, a focus on genetics, you guys hear about that all the time. Even if you don't have a genetic form of ALS, which is most of you, this research is relevant and matters, and a lot of exciting things coming from that. So how is it that we are kind of doing that in our clinic, and the mindset that we have? And I should say that one of my background is in another motor neuron disease where there now is a successful gene therapy. Spinal muscular atrophy used to be a fatal disease of babies. Now there is gene therapy that is basically keeping these babies from dying and developing the disease. So I'm not just trying to raise false hope here. That we actually have in humans done gene therapy on a motor neuron disease and have had clinical success on that. And we'll come back to that. So in the clinic, it's from large populations of people with ALS that we discover gene discovery, and that's not just in one clinic. Those are very collaborative types of things that are networked, and I'll talk a little bit about that. That gene discovery, if you have a gene that you associate with ALS, it does a number of things. One, it allows us to make those mouse models. It allows us to take cells from a person and take them to the lab, grow them, maybe turn them into an induced proliferative stem cell. You've heard of that before. And then a relevant cell type that we can study. Those are the things that then we can try to ask, what the hell's going on? What is it that one mutation, how did it, at that point we're like mechanics. The car rolls in, it doesn't work right, and we try to figure out what it is that's broke on it. And that is what then leads us to targeted therapies that then come back to the clinic. The other thing about gene discovery is that, and SMA taught us this, you don't have to know what the function of that protein is. You can design maybe a gene therapy to correct the error, and that is a treatment, and that's what happened in SMA actually. Okay, so let's go through this really quickly. So there are multiple gene therapy trials right now, and one of them that finished was this VALOR study. That's the one where it was found that drug Qalsody, which you probably heard about earlier today, was approved. It is the first gene therapy in ALS. It probably does have biological efficacy because it decreases the SOD1 gene expression, it decreases neurofilament, which is like smoke for the motor neurons. And some of them seem to be maintaining. We have, I think, 9 people now that we are administering this to, and it does seem to have an effect. I believe that we have really a truly disease-modifying medication now for that particular mutation. There are a number of other trials going on. We are one of a few sites doing another gene therapy trial. But this emphasis on genetics in the clinic has really, what 10 years ago I would say, was a little bit of a confusion as far as, well, how should we offer genetic testing? How should we do genetic counseling and so forth? And so and this is one of the things at Ohio State that occurred with the support of the ALS Association. Jennifer Rogenbuck, who's a genetic counselor and researcher, and Matthew Harms, who is a world-renowned geneticist at Columbia University of ALS Genetics, and I developed the ALS genetic testing guidelines. And so this has now been adopted throughout the country, and actually the European Academy of Neurology has adopted this. And so every person with ALS should be offered genetic testing. Of course, that's optional. You don't have to have genetic testing, but it should be offered, which was not the case in the past. And this has sort of implications for management that Jennifer, of course, is an expert of with the genetic counseling, but also for us nerds has the ability to identify people who are maybe candidates for specific kinds of selective gene research studies. So, so this is something that now happened here in Ohio State. We're changing the way people do their medicine. What's the other thing about the discovering genes? And that's the mechanism of disease. And Dr. Elbasiouny already illustrated this pathway, but I just want you to know that we have developed the first animal model of a KIF5A gene variant, and that has recently been published. This is Kelly Rich, my graduate student. All of these things, by the way, help young scientists get excited about ALS research. They get excited about going into this and saying, hey, this is something that I want to build my career on, is try to understand what is broke in that. So this is something that was sponsored by the NIH and also from philanthropy and other things. So just to sort of show you how this all works. The other thing that's happened here at Ohio State is we have a long tradition of asking our patients to volunteer cells from their bodies that we take to a biorepository. We take fibroblasts from skin cells sometimes, and we can turn those into relevant cell types in the lab. And there have been cells from people in Ohio that have gone all over the world now to go to, these are just some of the publications that model the disease that other scientists who might have a better idea than we have can use to study that stuff. So these are just some of the research accomplishments I've got. We actually have an X account now. Luckily, by the way, we have Carly and Cheyenne here. These are two of my team, the research coordinators. Please go up to them. They have all the pamphlets and all that stuff. But they're the ones who are in charge of that because I have no idea what all that is. But that is a way now that we are trying to keep people abreast of the trials that we're offering and also some of the scientific accomplishments, ALS-related things that are there. So it's kind of exciting. All right. So now, just to let you know, and this is something where we're happy to help and recruit also for your study as well. I think that it's all about trying to get the word out. We are part of the Healy platform trial that Dr. Deardorff was talking about and doing one of the gene therapy studies right now. It is a weird time in ALS. There aren't a lot of enrolling studies going on at the moment, but there's about to be a large number of them. And so for our studies, anyone can always email ALS Research at osumc.edu. That will go to us. And any questions about, hey, is this trial going? You can use that as sort of a navigation tool, and we're happy to direct people to other studies, that are within our catchment area as far as that goes. The Healy platform trial was already mentioned, and I appreciate Adam talking about that. It is one of these distributed studies, and we are one of the sites throughout the country that is enrolling, and we're one of the country's leading enrollers in that study. And just as Dr. Deardorff talked about, we are now through Regiment G, and they are going to be firing up for Regiment H, we hope, in the next couple months, as far as that goes. I wanted to share this. This is from the Healy group. Of course, we're very close to Mass General Hospital. Merit Sokovich is one of my mentors and colleagues. And so the Healy platform trial has these QRS codes that are also very helpful for people that goes to Mass General, but they can direct you to other things. And then I'm going to close now talking about some of the clinical studies that are going on. So when we think about clinical research, we often think about clinical trials, just as Dr. Deardorff talked about. Does this work? Is it safe, et cetera? But in that Buckeye tree, getting things to the lab, we also have to do those clinical studies. These are things that are not going to benefit the person who's volunteering. These are purely volunteer efforts. But we are very proud to be part of this all ALS consortium. And that's one of the things that I wanted to sort of announce. We have pamphlets about it and answer questions. We also are doing, of course, have the biorepository. And we, as a Muscular Dystrophy Association, a sponsored clinic primarily, we also are part of the MOVER registry. So any of these things you can go. And to be clear, a lot of people come to us for research and then seek and get their management elsewhere. So this is not about the clinical piece. This research is for anyone, no matter where they go, to receive their caregiving. In the case of all ALS, this is there's really two hubs: Mass General and the Barrow in Phoenix, and then there are spokes sites, and we're one of those sites. We're the only site in this region of the country, and we're working to try to get outreach to West Virginia and Indiana and Kentucky as well. What is interesting about all ALS, this is essentially a longitudinal, meaning we're going to follow people over time. There's a lot of biomarkers that are done. But one of the cool things about this and innovative things about this is a lot of things can be done at home. Because having to come into the clinic is a burden if it's coming in for a research thing. And so this are things, there's too much to talk about in this period of time, but this is what Carly is the lead for that, and she can answer questions. And then I wanted to kind of just finish this a little bit. There's a lot going on with at the NIH right now. And I think a lot of people, I've been getting questions about what's happening with funding for research, with changes that are happening in the government right now. Sometimes there's been some anxiety about it. I want to reassure everybody that everything is moving straight forward. And particularly at OSU, we've had reassurance from on high that, that this is not going to stop ALS research. But there are definitely changes. And I think it's important for people to be educated in what that is. So this all ALS project actually came from a law that was signed in 2021 called Act for ALS. And actually the ALS Association was extremely integral in doing the lobbying to make that happen. So this is a real win for the ALS community. And one of the results of that was this $38 million allocation for all ALS. That's how this consortium happens, and OSU is receiving some of that money. It results in jobs. It results in people being able to serve people who have ALS and get this going. So, if people have questions about that for the discussion, I'm happy to talk more about it. And also for enrollment or anyone, friends and family who are interested in it, then Carly and Cheyenne are here for that. So I'll wrap up and just leave things for questions. But there really is, I hate it when people come up and there's too much cheerleading about it. But there really is excitement that if not now, in the next generation, we are going to have disease-modifying therapies for ALS that might really change how we look at it. This is my lab here. Megan Baird is my postdoctoral fellow. And she is a recipient of the first research gift from ALS United. And I wanted just to thank again ALS United for that support and for recognizing that research is really kind of where it's at to get these things going. And then I have a number of the funding sources there for the research that we do. And then with that, I will stop and see if there are any questions, and then we can go on with our discussion. Thanks, guys. I went too long.
Strong work.
Yeah. Sorry if that went too long. Yeah.
You're fired. No, I think we do have a little bit left time if you guys aren't completely exhausted. This is a long day, but I think this is a really good way to end, right? I mean, this took, everything from learning more clinically about what's going on in your body to sort of giving hope as to what scientists are doing in the future. This is the future of ALS right here, from animal models all the way through to people with the disease coming in, getting medications, and we're studying them. So this is the whole gamut of research, and I think it's super exciting that all of this is going on here in Ohio. Very cool. So questions, I think we've got some microphones we can run around. If not, just raise your hand. We can ask some questions. You guys are fried? Probably. I am.
I'll ask one.
Yeah.
So, Dr. Kolb, you talked about how there would be a lot of studies that will start enrolling. So are those like phase 3 or, so when you think about those, is there any that you're more excited about?
Yeah, I know. That question is really interesting. So, a lot of people come to us, come to me, and want their trial to be done at OSU, and so I try to vet things a little bit. There's something based on the science part of it. So anything that we are doing, I think is a good shot on goal. The problem is, of course, we don't know because it's an experiment in humans that we need to do in order to know. But I'm particularly interested in being able to modulate gene expression in people who we know have those genetic forms, but most people don't have that. And so I'm also very interested in modulation. There are going to be things we're going to be ramping up soon on a study that is actually looking at neuroinflammation and looking at some of the local stuff that's going on there. So I think more general neuroinflammation at a cellular level and then the more targeted gene therapy kinds of things I think are interesting. And there will be gene therapies per se for people who do not have a known genetic form. And so that's what some of the research is showing is that even people who don't have a known genetic form, they may benefit from some of these other things.
So could you talk about that a little bit more? Actually, all three, because we talked about tofersen this morning,
Yeah.
how it's so specific, because
Yeah.
you have to have that code, and you have to have to give the other side of the code
Yeah.
to block it.
Yeah.
So how does that turn into better treatments in the sporadic people that
Yeah.
don't have those genes?
Yeah, I think it's going to be a very specific thing. For example, for the SOD1, the thing is let's just decrease that level of that message. So for that, no matter what variant in SOD1 you have, that will work to knock down that. But yeah, for sporadics, there's actually some evidence that there may be some benefit in cultured cells of knocking down SOD1 that are from ALS patients that don't have SOD1. But for Qalsody, I don't know whether that'll be it. I think that what's going to happen is that in the future, we're going to do a genetic characterization of everyone, including people who have non-genetic or we don't know what their gene is. And then we'll also probably do cell-based characterization. Just like you have 10 women with breast cancer, you do genetics, pathology, and then these 2 women get this treatment, 3, and so forth. So I think we're going to have more of a categorization of the biomarkers that are going to happen in the clinic so that then someone who would get a therapy that might apply just to them, specifically to them. I think one of the things that unifies people with sporadic disease is that TDP43, which is another gene that if it is mutated causes ALS. TDP43 though is mislocalized in all people with ALS, whether you have a mutation in TDP43 or not. And those are the gene therapies I think that are going to be for people without a known gene variant, those that are involved with the biology of that.
We love our letters. Just
Yeah, all these letters
right,
for
the letters
genes.
and everything.
Yeah.
TDP43 is a cool one, though. That's one to remember. If you're going to remember any letters, remember that,
TDP43 is a big
because you
one.
will hear more about this.
Yeah.
Definitely. Other questions? Yeah.
Is there anything in the pipeline that's specifically for C9?
Yes. Yeah.
That's a good question, yeah.
Great question. So, as I had on that slide, unfortunately, there was a gene therapy for people with C9, hexanucleotide repeat expansion, that ended up, it was a placebo-controlled, and the people on it actually did worse than the people on the placebo. So that was stopped. This is the experiment in humans. That's why we do clinical trials, because we just don't know until, it's done. Qalsody worked. This other one didn't. And so people are now trying to design similar kinds of things for, there's sort of a sense strand and antisense strand, so they're trying to look at that. The other thing, and actually the researcher who's at Ohio State, who I'm hosting now, she is the one who discovered that those hexanucleotide repeat expansions make these dipeptide proteins, little pieces of protein. and they get increased. And so now there are antibodies to go after those proteins for people who have C9. So people are bummed about that clinical trial, but it has not discouraged the pipeline as far as trying to attack that particular form.
Other questions? Marlon, I know I want to give you a couple minutes, but I think, Adam, Sherif, any comments?
One thing I can say is that this mutation-specific changes, we see them in animal models as well. So it's very impressive. One thing I want to say is that ALS doesn't have one scenario. This is what we see in animals. Some animals, they get the events, if I call them the disease events, sometimes the events are backwards. So sometimes in the SOD1 model, for example, cells die first before any symptoms. In the TDP43, symptoms emerge before cell death. So I do not think that ALS is a single disease. It's multiple diseases, but somehow they converge towards one destiny, one end. And it's really figuring out, and every mutation is a pathway to this end. So in order to be able to help patients, we have to figure out what changes this specific scenario this patient is going to go through just because of these, some challenges like the C9, for example. The animal model of the C9 is not that perfect. So this is making it challenging for us in the lab to really study that. The SOD1 is the most established, but the SOD1 is only 2% of the patients. Interestingly, the SOD1, well, in TDP43, TDP43 mutation, I'm trying to use like simple language, like, protein aggregates. They are in 97% of patients, but there are only 3% that do not have them. Guess what? SOD1 is among the 3%. No patients or the animal models have TDP43 inclusions. So different scenarios. So the animal model that we have been studying the most is an exception. But it's an exception in a good way because we're trying to understand ALS in a different scenario. So it's really, I think it's good that it's a rare disease, but it's making it difficult for us to study because, to have rigor, you need to have a large sample, right? We don't have this large sample in humans, but we have it in animals. But our goal is to study ALS, to treat ALS in humans, not in animals. So you see, there are many things that we have to figure out and assumptions to make. But I feel the critical mass, I agree with you that I think the critical mass in ALS research is really big at this moment, and I'm very optimistic about having something developed hopefully in the near future. At the time when I decided to study ALS, we were very, very, very few, but it's not the case anymore.
Yeah.
I would say just remember, when you don't see us, we're in the lab, and we're working, and we're doing stuff, and we will never stop.